Okay, welcome back to the final AP Bio review. Last video we talked about enzymes, mitochondria and cellular respiration, and photosynthesis in chloroplasts. Today we're going to talk about cell division, specifically mitosis and meiosis. Okay, so I'm going to start us off with a little tip when it comes to memorizing all these things that you are listening to me talk about or anyone else that you watch. When you're writing notes, the best color to remember things in is in blue, which is just a small little fact that might actually help you a long way. So, if you're watching this video, follow along with me, write some notes, and if you are, write them in blue pen. Okay. Let's talk mitosis and meiosis. First, there is some quick vocab that we have to get across before we get into it. Chromatin is a more compact DNA, but it's still relatively loose. A chromosome is an even more compacted chromatin, and that is what's used for cell division. It looks like this kind of thing. A sister chromatid is just one. If a chromosome is two sides, a sister chromatid is just one side of that. A centromere is what attaches to the sister chromosomes to pull them apart. And it does that through spindle fibers that it will, pull them with. A kinetochore is where the... Oh, whoa, I totally lied. Okay. So a centromere is what holds these sister chromosomes together, right? It's this little itty bitty thing. You can't even see it on my drawing, but it's this little thing right in the middle of your chromosome. And the kinetochore is where the spindle fibers attach to the sister chromatids to pull them apart. Somatic cells are cells that have two sets of daughter chromosomes and underwent mitosis. And gamete cells are cells that underwent meiosis and only have one set of chromosomes. So that's your sperm cells, your egg cells, etc. There are 5 stages to mitosis. There's interphase, prophase, metaphase, anaphase, and telophase, right? And these stages are what, you have to remember to know what you're doing on the AP test. These ones, definitely. If you ever see a mitosis or meiosis question, these will be in there somewhere. So you have to know where each step is relative to each other. Starting with interphase. Interphase is the longest phase, and it looks like this. So this is your prophase, your metaphase, your anaphase, your telophase. This whole thing right here is interphase. Right? You have 3 separate stages in this phase, and the first stage is the G1 phase, which is growth part 1. Cells are growing, making sure it's all good for cell division. If everything is not good, then it will go into G0, which means okay, I'm done, I'm in retirement, I'm getting a vasectomy, no more babies, or no more cell division. S phase is the synthesis of new DNA, right? You're just copying all that DNA, getting it ready. And then after that, you have the G2 phase, which is growth part 2. Your cell continues growing, and it's making sure that it's all good. It has its necessary proteins, organelles, all that kind of stuff. And once all of this is done, it can go through the rest of mitosis. And speaking of the rest of mitosis, here is the rest of mitosis. So we start with prophase, which is when the chromatin condenses into chromosomes and the nucleus starts to dissolve, right? And you have spindle fibers that start reaching for those chromosomes. And in metaphase, the nucleus is fully dissolved and the spindle fibers attach to these chromosomes. And they line them up right in the middle at what is called the equator. And it pulls them apart in anaphase. In anaphase, sister chromosomes are pulled up, or sister chromatids are pulled apart by spindle fibers and are pulled to opposite poles of the cell, and the cell gets just a little bit longer. In telophase, the spindle fibers continue pulling away, the nucleus starts to reform again, and chromosomes go back to being chromatin. And telophase is almost made out of two parts, which is the first one was telophase, and the second one is cytokinesis, which is what you're seeing right here. The cell starts pinching itself in to just like split apart like that into two separate cells, two separate daughter cells. And that's what it looks like in animals, and it is just slightly different in plants. So in plants, a cell wall is put in between the two cells in order to separate them instead of, pinching them down. That's the only difference. So as you may have seen in my picture of the interphase, I have these little checkpoints right here at the end of G1, the end of G2, and right at metaphase. So these checkpoints are if something goes wrong in the cell while mitosis is happening, it will either enter G0, as I mentioned, and that's when it no longer can do mitosis, or the cell will kill itself in a process called apoptosis. There are 3 checkpoints where a cell does its checkup. In G1, the first one is it's asking, all right, am I big enough? Am I healthy enough? Is there any DNA damage? And if the conditions are not right, it will go into G0 or, kill itself. G2 is the DNA copied correctly. If not, try to fix it. If not, if it's like beyond repair and you can't fix it, then apoptosis. Okay, the next one is in metaphase. Are all the spindle fibers lined up and attached correctly during metaphase? If not, apoptosis, right? And the way that this is checked is through cyclin and CDKs. So cyclins are used to as a check mark saying, okay, you can keep on going. And CDKs, or cyclin-dependent kinases, need kinase in order to work properly as a protein. So if you remember, kinases are those enzymes that help release phosphates. And cyclin, being something that this kinase is dependent on, almost acts as a cofactor, right? It's that second key that you need to get this thing working. And it is those things that, help the cell push on through with these phosphates signaling to the rest of the cell, okay, keep on going. Now we're going to be briefly talking about cancer. Cells become cancerous when too many genes telling it to stop are turned off, and or too many genes telling it to go are turned on, when both of those shouldn't be. So an oncogene is like a gas pedal, right? If it sees a green light, it will press on the gas pedal and it will go. A tumor suppressor is, okay, do I see a red light? And if I do, I will press the brake pedal and I will stop. If a mutation is bad enough in the specific place, it can be like cutting the brakes in the tumor suppressors or putting a brick on the gas on the oncogene. So even if there's a red light, it will just keep on cruising through that intersection. Or if it sees a red light, it won't be able to stop because the brakes are cut. There's 2 kinds of cancerous growths that will occur from this. You have your benign tumors, which is when a cell kind of freaks out, but it won't spread at all. It'll be in a contained little lump. While a malignant tumor is the one you do want to worry about, right? This is when the doctors get called. It's when a cell freaks out and it starts spreading out to different tissues, which is, when it becomes really bad cancer. Okay, now let's talk about meiosis. So meiosis is cell division for gamete cells, which are like egg or sperm. It has 2 stages, meiosis 1 and meiosis 2. So in prophase 1, chromosomes are duplicated, the nuclear membrane dissolves, and the spindle fibers start forming and crossing over happens. Well, what is crossing over? So in crossing over, you have 2 chromosomes, one from mom, one from dad, that are exactly the same. And they get right near each other, and they get on top of each other in what's called a tetrad and start swapping DNA segments. And the reason it does this is to make the offspring more variable. So not only will you have a 50% chance of getting it from mom or a 50% chance of getting it from dad, it might be 51, 52, 48, 49. It'll be switched around a little bit. And in step 3, as I've listed right here, homologous pairs get off of each other and now have some segments from each other traded. And all of what I just mentioned happens in prophase 1. And prophase 1, same as prophase in mitosis, basically, right? Your nuclear membrane is still being dissolved. Spindle fibers are still coming towards those chromosomes. It's basically the same thing. So metaphase is just a little bit, or metaphase 1 is a little bit different from metaphase in the sense that homologous pairs line up side by side. So these two chromosomes will get up right next to each other and they will undergo random orientation. So there is an equal chance that mom will end up on the left side and dad will end up on the right side for each separate chromosome. Okay, so it's really just chance what goes where. In anaphase and telophase 1, chromosome pairs are ripped apart in anaphase, and in telophase, the nuclear membrane reforms, becoming two diploid cells. Sister chromatids are still together, though. Okay, these X's stay condensed for meiosis 2, right? And cytokinesis still happens. I just didn't mention it here. That's what the pinching is. So meiosis 2, I drew it a little bit smaller because it is basically what mitosis is, with, prophase 2, your nuclear membrane is being dissolved, metaphase 2, they're being lined up, anaphase 2, they're being ripped apart, and telophase 2, chromatin starts devolving again. And after this has all been done and dealt with, you end up with half the amount of chromosomes that you would usually have. And there are 4 daughter cells instead of 2 like in mitosis, right? So whatever happens here, this creates 2 cells. And then these 2 cells will split and do 2 more cells through meiosis 2. And that's what leads to you having 4 daughter cells. Sometimes in anaphase 1, the homologous pairs won't separate, leaving one cell with an extra chromosome and another one missing one. And this is how Down syndrome or triple X happens, which is scientifically called non-disjunction, which is when they don't leave. That is everything for meiosis and mitosis. That was a pretty quick video, relatively. If you like that one, I would really recommend subscribing, as more are on the way. And in the next video, we are going to be talking about genetics. Okay, I will see you there.